Good afternoon, and welcome to the DeWitt Stetten Jr. Lecture. I am John Lorsch, Director of the National Institute of General Medical Sciences. I want to begin by telling you briefly about the distinguished scientist this lecture honors. Dr. Stetten spent most of his career here at NIH, serving in 2 institutes as well as the Office of the Director. He was probably best known as the NIH Deputy Director for Science, but before that, he was the 3rd director of NIGMS. In recognition of his many contributions, both within and outside of NIH, Dr. Stetten had the rare distinction of having not only this lecture named after him, but also the DeWitt Stetten Jr. Museum of Medical Research, which is also known as the NIH Stetten Museum. Our speaker today, Romy Amaro, joins a distinguished list of Stetten lecturers. In her work, she applies increasingly sophisticated algorithms and computing power to drug discovery. Her goal is to find new ways that small molecules can restore function to affected proteins in diseases such as Alzheimer's and cancer. As you will see, Romy uses multiscale dynamic simulations of protein structures to predict how proteins move and change shape. Her work on the cancer target P53 revealed a novel pocket in the protein's core that is receptive to binding by small molecules. This discovery clarified the mechanism of action for a compound currently in clinical trials. The work also became the basis for Actavalon Incorporated, a startup company that Romy founded to develop P53 reactivation drugs. Romy has a BS in chemical engineering and a PhD in chemistry, both from the University of Illinois at Urbana-Champaign. She is now a full professor in the Department of Chemistry and Biochemistry at the University of California, San Diego. NIGMS has actually supported Romy's research since she was a postdoctoral fellow at the University of California in San Diego, beginning in 2006. Please join me in welcoming Romy to give this lecture. Thank you very much, Romy.
Okay, are we live, this mic? So I'll go back one slide. Can you all hear me? So thank you. Thanks for that kind introduction, and thank you for the invitation to give this lecture. I am really super honored to be giving this lecture here today. And it's funny, for some reason, it feels a little bit like a homecoming, even though I didn't spend any particular length of time on this campus, but it's really great to see friends and colleagues who I've now known for a number of years. And I'm really excited to tell you today about how we are using computing and different types of biophysical algorithms to inform on drug discovery, essentially. And so the title of my talk today is Computing Cures Discovery Through the Lens of a Computational Microscope. And so I like to start out my talks by sort of just emphasizing what an exciting time it is to be a computational chemist. That's what I am. I'm a computational biophysical chemist by training. And really what's so exciting is that computing is really transforming chemistry and biomedical research. And so what I'm showing here on this slide is the available compute power, this mega lightsaber type laser pointer. Anyway, I'm showing the available compute power as a function of time. And so this is, what I'm plotting here is ever since the early 90s, the US invested in these, what they call the supercomputing sites. The first one came online in 1993 and had 12 CPUs. At the time, that was just fantastic. And what you can see is that every 4 to 5 years since then, we've increased our compute power by about an order of magnitude. And so these machines have grown in size from 12 CPUs up through today, where we have machines that have multiple hundreds of thousands of cores. And in addition to that, are also accelerated by graphical processing units. And why we care about that is that it turns out that the same types of computational hardware technology that is useful for driving the huge industry of video games and rendering video graphics realistically in real time, it turns out that these chip architectures actually are highly amenable to the types of scientific computation that we want to do in the space of computer-aided drug design and biophysical simulations. And so I would say computing coupled with advances in data science, which I know NIH had a big effort in BD2K, and in general with the rise of big data and data science related objectives, we are in a unique position to really harness computing power in new and exciting ways. And so I would say computing is really transforming how we do chemistry and biomedical research. And it used to be that we would think of chemistry, and even today, if we think about chemistry and biomedical research, we think generally of wet labs, of people wearing white coats and glasses. And there's a whole another side to it. And I would say now, for example, what we might do is actually get data from one of the beautiful microscopes that I know many of you here are familiar with, for example, structural data. This data could be taken at the microscope, and then, for example, on the UC San Diego campus, which is my home campus, we have a whole infrastructure related to very fast data transfer. So we have basically these data freeways that pass very rapidly data that's taken from the microscopes to, for example, a supercomputing site, which we actually are lucky to have at San Diego. So there's supercomputers there called, one example is Comet. So the data goes from the microscope directly to the supercomputer where they do various refinement, and then we can pull that down back in the lab and start to look at, for example, some of these structures in really beautiful detail because of the processing of these algorithms. Then what we might do is we have, say, a molecule or a supramolecular assembly, and then we can augment this data with various other data sources from different community resources. We might do that to build an even larger system that we then put back out on the computers, such as Blue Waters, which are these large, very large scale petascale machines. And then once this computation is finished, we can pull it back into the lab and do various types of not just sort of regular or standard interaction with the data, but we can also do things like virtual reality and so forth, and there's certainly plenty of people who are developing that. And so I would say that nowadays computing has transformed the environment of science into something that combines experiments, simulation, networking, and expertise as sort of a science super facility. I also think that computing has tremendous potential to transform who is doing chemistry and biomedical research. And some of you may recognize this famous photograph from a European conference of physical chemistry circa the 1920s. I'd say that was the face of sort of physical chemistry then. And this is the face of physical chemistry, or it's changing to engage a much sort of younger, more diverse, not just younger, but diverse audience of all types of backgrounds. And so an example here actually is a student who came through the center that I run and my lab, who as a 17-year-old was able to discover, to use the same types of approaches that I'm going to tell you about today in high school, to discover a novel compound, that in a chemical matter that actually was active against an enzyme in influenza called endonuclease. And so he actually had, even before he graduated high school, he had a first author publication in a very good peer-reviewed journal. And he also happened to go on and win the Google Science Contest, the Siemens competition, collect over $500,000 in prize winnings, and get a trip to the Galapagos. But in any case, it's becoming much more accessible nowadays. And so the other sort of, before I launch into sort of the more technical aspects of the talk, I just wanted to emphasize my own personal view, which I know many of you here share with me, and that is that I believe that the true research breakthroughs are going to occur at the interface of observational and simulation science. And these are going to be the breakthroughs that solve our most pressing challenges facing humanity. That's from environmental and atmospheric chemistry, if I can say the words quietly, something called climate change. Also developing new energy and materials to address issues in sustainable energy. And what I'll talk to you mostly about today are tremendous advances that have been brought on by computational approaches that are giving new insights into biology and medicine, and in particular, developing new therapeutics. Okay, and so I am particularly interested in using an approach in my lab and much of the resource activities that we have uses an approach called molecular dynamics simulations. And well, I wouldn't be a physical chemist if I didn't show you at least one equation, but I promise I won't show you very many more than that. And so the idea here, one of my former mentors, Klaus Scholten, sort of coined the term of using these approaches like a computational microscope. And in fact, I think that's a very good way to think about what this technology can do for us. So one of the things I love about it is that it's a highly interdisciplinary approach. It combines concepts from chemistry, physics, mathematics, and computer science in order to address new and exciting questions in the space of biomedical research. And so what we do is we basically, we're talking about molecules and cells, and so we have, for example, proteins or receptors, and we're approximating our system, we're modeling our system at the atomic level, and we're basically approximating each of the atoms in our system like as hard spheres. And so once we do that, we can define what we call a potential function, here called U, that has different terms representing the atomic dynamics and interactions that occur at this small scale. And so every 2 atoms that are connected in our system form a bond, and they have a harmonic spring constant here, so they have a term in this function. Every 3 atoms that are connected form an angle. Every 4 atoms form dihedrals. And then we have what we call the so-called non-bonded forces, which is basically electrostatic interactions, sort of longer range interactions, and also close range interactions like van der Waals potential. So once we define this, and what we call a force field to go with it, all we're doing is basically integrating Newton's equation of motion over time. And so what we do is typically we start with a high resolution crystal structure where we can get, or it could also be an NMR structure, where we get the positions of the atoms in our system. We start with this initial system, we integrate a certain time step, and this time step is very small. So when we do this integration, we're limited by the fastest scale motion in our system, which is the carbon-hydrogen bond stretch. And so that means we can only take a time step of 1 to 2 femtoseconds of biological time, which is very, very short. So we have to integrate millions and billions and trillions of time to actually build up a dynamical trajectory and get to the types of timescales that are useful for us. So we start with one particular structure, we integrate time t, we get another structure, we integrate again, we do this, and we end up with an ensemble of structures that describes the time-dependent trajectory or dynamics of the system. Okay, and so why do we care about that? I'm going to tell you a story about why we care about the time-dependent dynamics of our system. That story involves a molecule that probably many of you in here are already familiar with. It's something called P53, which has also been called, many people refer to as the guardian of the genome. So P53 is a central tumor suppressor protein. It basically senses various types of cell damage and then upregulates pathways related to essentially cell death or cell senescence or growth arrest. And so in a normal functioning cell, the P53 will basically sense the damage and then upregulate all of these factors that help kill the cell. But what happens in over 50% of human cancers is that P53 has a single point mutation, and this point mutation renders it inactive. And so what happens then is that this mutated P53 does not, so the cell is damaged, and then P53 is no longer able to upregulate all these pathways related to growth arrest and apoptosis, and therefore the cells just continue to proliferate. And so there's been a lot of people studying this protein for many years. It's one of these sort of central cancer hubs. What we focused on initially was something called the core domain or the DNA binding domain. So P53 is actually a fairly long protein, but we were just focused on what we call the DNA binding domain. And what this image here is showing is the frequency of P53 mutations that have been found in tumors, in patients. And what you can see is that the mutations can happen in a lot of places, but there's really a predominance of about 6 amino acids if you look at the whole sort of, the entire patient population. One of the things that's really interesting about these mutations that I'm showing here in this lower left-hand panel, so in green In the green ribbon, this is what the DNA binding domain looks like. They have many high-resolution structures of the p53 protein. This is represented just this core domain down here. So you can see these little red dots. These red balls are actually indicating where in the structure these cancer mutations can occur. And so one of the things that hopefully jumps out at you is that they're not all localized to one particular place. A mutation can happen in this area of the protein, in this area, or down here, and somehow still it's rendering this protein inactive. Another interesting thing about p53 is that it has the ability to adopt a secondary mutation, and sometimes a tertiary mutation, that can actually restore its activity. And these are so-called cancer rescue mutations, and I'm showing those here with the blue spheres. And also you can see that these cancer rescue mutations are sort of well distributed throughout the structure. So just to sort of recap how it works is that when you have wild type or active p53, everything is fine, everything is working. p53 can adopt one mutation that renders it inactive. These are the so-called cancer mutants. And then it can actually reactivate itself by adopting a secondary point mutation. And it turns out that when these single point mutations happen, one of the main reason we now know why that actually renders p53 inactive is because p53 is essentially marginally stable at physiological temperatures. And so these point mutations just shift the folding landscape ever so slightly and create essentially, they sort of disrupt the folded state of the protein. And the rescue mutations actually restore that stability and so can confer function again. So a long-standing dream of cancer biologists, as you can imagine, has been to develop small molecules that are actually able to rescue p53 in patients. And so the idea would be that you had this inactive p53 cancer mutant, you would dose the patient with some kind of drug, and that it would reactivate p53 and either regress tumor development. And so there were a number of studies in 2006 and 2007 actually showing that restoration of p53 function in vivo actually led to tumor suppression or tumor regression. And so people really sort of, I think at this point, started to become more convinced that this was actually a viable target. After a couple of decades of study, however, if you look at the literature, there's only a small handful of molecules that were known to reactivate p53. And so I'm showing these molecules here. And one of the things, one study in particular, which was published in 2009 by Fairst and co-workers, Alan Fairst and co-workers, presented this Prima-1 compound and possibly some of its degradation products that could be, that were showing actually p53 reactivation activity. And one of the interesting things was that they knew that these guys, if you're a chemist, you'll notice that these guys will covalently attach onto cysteines. These are covalent compounds. So they knew that they were covalently attaching to the p53 binding domain, but they couldn't figure out exactly which cysteine was responsible for the reactivation activity in vivo. And so we started studying this system. We became very interested in it because it was known to be a flexible target. And so what I'm showing you here is that DNA binding domain, the core domain of p53, and the wiggling and jiggling, that's the wiggling and jiggling of the atoms as predicted by these molecular dynamics simulations. So what you're looking at is a movie of only about 20 to 50 nanoseconds of sort of real biological time simulated in a water environment of what the atoms in this protein are actually doing. Okay, so and I'm showing some of the protein in purple, and I'm sort of highlighting one particular area in what we call in green in a different representation, in a surface representation. And I did that to highlight the motion and mobility around this is a cysteine residue that I'm showing here. And what you can see is that there's this pocket, there's a lot of dynamics happening in this area, and there's a pocket that actually opens up and reveals itself in the dynamics. And we became very, very interested in that. So if you look at as I mentioned, this has been studied for a long time, and if you look at the PDB, the protein data bank, you can find over 100 crystal structures of the DNA binding domain of p53. And almost every single one of them looks like this in the area of that particular cysteine, cysteine 124. Basically, the cysteine is tucked back and occluded from solvent and appearingly not able to be covalently modified by any small molecule compounds. But what I just showed you in the trajectory on the last slide, we predict there's a lot of mobility in this pocket, and in fact, this is an example structure from the molecular dynamics, or MD trajectories, that indicates and shows an open pocket. So we were very excited to see this. And when we find this pocket, and then we used a technique called computational solvent mapping, which basically allows us to assess the druggability of any site on the protein, but in particular we were really interested in this new site. And the way that it does this is you take your crystal structure or whatever structure, it could be an MD structure of your drug target, and you flood that structure with different organic probes. And then all we do is assess the interaction energy of these different probes with the surface of the protein, and it indicates to us where on the surface of the drug target, of the drug receptor, small molecules might have a propensity to bind. So we found this pocket, we predicted that it's druggable, and then we carried out a series of small molecule docking campaigns against this pocket. And what we found, so this is actually a sort of a schematic depiction of our workflow. So we have different, what we call the receptor ensemble. So this is our drug target in our different pockets. We start with the X-ray structure and the MD structures. And then we use something called AutoDock-Bina, or a docking program, to basically try to see which small molecules will actually fit inside that new pocket. And when we tried to predict which would be sort of the highest or best binders. And so, so what this means is that if you're an experimentalist, obviously instead of having to test 2,000 compounds or 10,000 compounds, we can give you, our computational folks can give you a much smaller set of compounds. In this case, we gave them 24 compounds. And we were crossing our fingers hoping that this set that we gave them was actually enriched for binders, as opposed to just choosing randomly or having to test the whole library. So what we found, we did find one molecule in this rather small campaign. We found one molecule called stictic acid, which I'm showing here, that actually showed dose-dependent rescue in mammalian cancer cells. And here we were testing different transcription factor readouts. And the other thing that was really interesting that we were able to do is through mutational studies, through mutational analysis at that particular cysteine position, we were able to show, in fact, that that this particular cysteine 124 was, in fact, the position that is modified and reactivated for the compound that's currently in clinical trials. So we were both able to discover a novel reactivation compound as well as rationalize a compound in clinical trials. And so, as you can imagine, then we were quite excited about this, and so we significantly expanded the campaign to find small molecules. So again, we focused in on one of the most productive structures, this with the open pocket, and now instead of searching against hundreds or thousands of compounds, we're looking at millions of compounds. We also tried different data mining approaches in collaboration with some computer scientists, and then passed it along to our experimental collaborators. And now what we were able to find were multiple dozens of compounds, in fact, that reactivate P53 in various cancer cell lines. And we were able also to find compounds that reactivated multiple mutations. So it could be not just against the R175H mutant, but also the G245S mutant. And so one of the cool things, so this work actually formed the basis of the formation of Actavalon Incorporated, which I'm required to tell you I'm a co-founder of, I hold equity interest in, and I'm on the scientific advisory board of. This now just moved over to J Labs and received another series of funding. But one of the really cool things, one of the reasons, I think the major reason I like to tell this story is not only possibly that we could develop compounds that might actually make a difference one day for patients, but also that it's a very nice example of how the power of a good model. And that in 6 months, using this approach, we were able to discover more reactivation compounds, essentially, than in all the previous 20 years combined. So that was really cool. And I'm sure if you know anything about cancer, it's a devastating disease. If you live in the United States, you have a 1 in 3 chance, I believe, of being diagnosed with it in your lifetime. And so it's going to take a lot more than just one lab or a few labs who can do this. So one thing that is very near and dear to my heart is the development of software and methods that others can also use. So if this is a good method, let's put it into the hands of others so that they can also, with their cancer target, also develop novel compounds or have a better chance to find them. And so to do that, we partnered with NVIDIA, who's one of the GPU chip developers, one of the main ones, and we were awarded from their Compute the Cure Foundation. We also partnered with the developers of Kepler, which is a workflow tool, and created a series of workflows that sort of help to decomplexify or demystify the drug discovery process. So the whole process of from when we get the crystal structure to actually coming up with a set of compounds that's going to be useful in the lab, there's well over 100 or 200 steps involved with that, with many different decision points. And so the generation of these workflows is one way that it's not only computing power, but also the power of data science to help others also access and use these models. And this is probably way too much information, but I just wanted to show so that typically these types of experiments require scientists to run and sort of cobble together many, many different scripts and to develop this sort of recipe. And what we do is we basically encode these scripts in sort of graphical user interfaces so that becomes much easier to run. And we also develop these workflows so that they can easily connect to and run on the many different computational platforms that researchers now have access to. So not just on your desktop with a single GPU, not just on the cluster you may have in your lab, but also on resources like Exceed, which are the supercomputing sites that are sponsored mainly by the NSF, and also getting these up and running on Amazon so that any researchers can basically get up and running with it. It also makes it a lot easier to train students in all the different steps that have to occur. Okay. And so that's what we've been able to do and really try to push forward at the really sort of the molecular scale. But one of the things that we're very interested in is sort of pushing the envelope and helping to address what we think are some of the most exciting and challenging areas of biomedical research. And I would say, from my vantage point, is we have a lot of tools to if we think about sort of the scale and the spectrum of drug action, if we really want to understand how the mechanisms of a single compound, chemical compound, can interact with a receptor and how that plays out over the hierarchy of scales, we need to have methods that allow us to inform how information from one scale is passed to the next. So we have good methods, for example, to investigate molecular structure and dynamics, but where we've been limited is in really understanding how these drug targets are actually situated in their subcellular neighborhoods, how these neighborhoods fit into cells, and how the cells interact in ecosystem-like communities of tissues. And so that means in order to do that, we really need to have a range of approaches that allow us to interrogate robustly across scales stretching from the angstrom at least to the millimeter or beyond, to the whole organ, in fact, and for temporal scales from the very small motions, the femto and picosecond dynamics at the atomic scale, into millisecond and second and lifetimes. And so that's a big challenge. But ultimately, if it's one that we do correctly, we'll really ultimately, hopefully, be able to understand truly the molecular and chemical mechanisms that underlie disease. And so I would say that it's not just the computing that's so exciting, and that's what I sort of stressed at the beginning, but we're also very excited about all the new sources of data, in particular structural data, that we has had tremendous advances thanks to a lot of technologies, but mainly advances in direct detector technologies. So we have at the single sort of molecule scale, we have beautiful data from electron crystallography. Of course, I think many people in here are already familiar with cryo-electron microscopy. This gives us images about supramolecular assemblies. We have approaches like electron tomography that basically lets us start to look at things like cells and organelles. And we also have things at the next scale like serial section EM tomography, where we can take sort of epoxy resin embedded samples and understand how these macromolecular structures are situated inside their subcellular neighborhoods. And at the next level, even with things like really trying to stretch technology, looking at things like serial block EM, where they put an ultramicrotome inside the SEM. And so here we have these sort of tissue samples, and they'll image the top layer, use a jeweler's diamond knife to shave off a very thin layer of the top of that, and then image another layer and do that again. These guys run for weeks at a time. They generate petabytes of data. And you get these beautifully detailed sets of data that have hundreds of thousands of structures in a single data set. So the dream is to really try to put this, or a dream for me and for people who I work with, is how do we take all this beautiful data, and it's not just structural data, also proteomics data, et cetera, and really try to make sense of it in the complicated network of cells. And so the last part of my talk, I'm going to tell you about our efforts to really drive and push the boundaries of molecular simulation into the mesoscale. To do that so that we can understand the drug target in its realistic environment. And to do that, we're faced with a lot of technical challenges. At least this includes things like multimodal data integration so that we can build structurally realistic models and detailed models. And the other issue that we're faced with is as we build these larger systems, it takes longer to simulate them. We need new approaches or bigger computers or both in order to actually extrapolate out to the timescales that are of interest to a lot of these problems. And then the third branch that really is a technical challenge is complexity and reproducibility. That when you build, it's hard enough to build a good model at one scale. When you start to connect them, now you sort of are combining the challenges and problems of both worlds, which can be difficult. And so I'm going to talk to you about our new innovations to try to address these challenges. So the first story I'll tell you is about P53, because I just told you about it. So this gives us a little bit of continuity in the storyline. So I talked to you in the first part of the study of this story how we had focused on the dynamics of this purple region here, the so-called DNA binding domain. That was really all that we were focused on before. And even just that turned out to be very successful for us in finding small molecules. But there's still so many questions about how P53 actually works and what it looks like. So this is this is a model of the full length P53. You can see it has multiple kinds of domains, including some disordered regions. And we basically used integrative modeling to develop a larger scale model of P53 in complex with different DNA strands. So here we can look at different response elements, so the P21 response element, the PUMA response element, and also non-specific DNA. And so now we're talking about, instead of just looking at a single protein that has maybe 40,000 atoms, we're talking about looking and investigating the atomic level dynamics of systems that have on the order of 1.5 million atoms and have much more complexity in terms of their dynamics. So P53, this is actually that long stretched out protein I showed you before. This is actually, that's actually a tetramer, so there's 4 copies of those, and they come around and grip the DNA in various ways. And so this integrative model of the dynamics is going to help us inform on the atomic level information about what's happening in terms of the movement of the system. And then we can do things like compare our model and simulated density maps to maps that they have from cryo-EM to sort of check how well it matched and make sure that we're not completely off. And there's many different ways, of course, of validating these models, and we try to do as many of them as we can while we're developing and analyzing these models. Okay, so again, so why do we care? Well, one of the interesting things that we found out when we moved to studying P53 in its more realistic environment is that P53, as I mentioned, it's a tetramer, and so you can see here's one copy, one domain, another domain, here's the third domain, and a fourth domain. Depending on which domain you are, if if we're personalizing the protein, the loops actually can interact in different ways with the DNA. And what we found was that these interior loops actually lock the molecules, the two subunits of p53 that are shown here. They actually are persistently interacting with the DNA, such that that pocket that I showed you before, that novel pocket that looked like it was constantly opening and closing, it turns out, we think, that when p53 is actually in its more realistic form, that this interaction with the DNA basically locks the pocket in an open conformation. So in fact, maybe that's a reason why some of these molecules have really good access to the site. The other thing that we've been studying is just, we're very interested just in sort of the molecular recognition of the system, sort of, which is more of a little bit of the basic biology question. But what we found was that depending on the exact sequence of the DNA that p53, the transcription factor, was bound to, so for example, if it's bound to p21 or PUMA, which are known response elements, or if it's just binding a non-specific part of the genome, we actually see that p53 changes shape. It goes from, in a non-specific region here, it adopts an asymmetrical conformation. When it comes along the DNA and it hits a response element, it adopts a symmetrical conformation, and when it does that, it has a whole different way of gripping the DNA. We define something that we're now calling a grip volume. If p53 is actually more symmetrical, then there's room and contacts being made with the DNA that is different. So we suggested this as a mechanism of recognition for p53 for response elements versus nonspecific DNA. And then we further extended that recently by looking at different mutations. And what we see is that for these mutant p53s, they adopt that asymmetric conformation, which is less able to make contacts and grip the DNA, even when they are bound to a response element. And so we're putting this forward as sort of a general mechanism for how these transcription factors are acting, or this one in particular. Okay, so that builds us to the point of relatively simple molecular assembly, but still sort of pushing the envelope on what people know, but we want to do more than that. And so we're developing at the center, at the National Biomedical Computation Resource, which is an NIH-funded P41 resource, we are developing a tool called CellPack. And so many of you might be familiar with the beautiful images made by David Goodsell at the Scripps Research Institute. They sort of show the intricate molecular complexity of these biological scenes in ways that, are just in some cases really mind-blowing. So what we're doing at the center is taking a tool. So this was a tool that was originally developed at NBCR, more from a graphical, for an artistic or videographic type representation. What we want to do is take that same tool, but actually build structurally realistic models and then connect those models to different types of biophysical simulation platforms. And in doing so, this is going to, we think, it's actually revolutionizing essentially the way that we carry out and inform on molecular simulation. So what we can do with CellPack is basically take data from, for example, microscopy, tomography, et cetera, and define different surface containers. In this case, what you're looking at is HIV. And then if we know the different molecular ingredients that belong in this particular system, then in some cases we have high resolution structures of those different molecular ingredients. In some cases we know their stoichiometries and have other constraints about their position and location. Then we can use CellPack to basically construct these highly detailed atomic level models, again at the mesoscale. This tool has been widely downloaded already since our first release in 2016. And actually I should say, so this is Art Olson. This is a tool that we co-developed at the center with Art Olson and Michelle Sanner, who sit right next to David at the Scripps Research Institute. So, CellPack is, in fact, a cell-centered, data-centric modeling environment that takes as input data essentially any data that you can feed in. Not only structural data, but also you can have fluorescence data, we can take proteomics data, and you can mine the literature for any kind of constraint. And then basically we parameterize, the user parameterizes different recipes that constrain the model system. And so, for example, in this image of a bacteria, we basically define, okay, these are the interior ingredients shown here. We define the space that they can occupy. We define surface ingredients and the space that they occupy. And then CellPack will very rapidly, just using your desktop, stochastically populate a series of models that are all in agreement or constrained to the particular recipes that you've put in. So one of the cool things about this is that it used to be that just to build one system at this scale would take you months or even years. And now we can not only build one system, but CellPack allows you to build multiple systems in minutes that are all in agreement with that data. So the view here is that ultimately it's going to allow us to get a better sense of and address biological heterogeneity, which is like a very big challenge for a lot of the things that we want to study. Okay, and so from a tools development standpoint, what this means is if we want to actually study this at the atomic level, we need new tools that allow us to actually build these systems at the atomic level. So we had to build something, for example, called lipid wrapper. And here what we can do, so lipid wrapper for the membrane, or the lipid bilayer in cells all over the place. So what we do is we can define, again, from tomography or any type of input data, we can define the surface that we want to wrap in these lipids. And then we basically tessellate that and populate these different triangles with atomic level models of lipids. And there's a lot of work that we have to do to remove clashes and get it to run successfully, but we have. And what this allows us to do, tools like this allow us to, for example, build whole influenza virus structures. And so this is actually a collaboration that I've had ongoing for some years with Alistair Stephen, who's in the audience. So what we've done here is, it used to be that molecular simulators, or people like me, we would just study these single glycoproteins in a lipid slab. So if you wanted to study these glycoproteins, you would just take this square planar lipid, insert your protein, and then simulate it like in an infinitely dilute water box. What we can now do is actually, with these tools like CellPack and Lipid Wrapper, we can take that tomographic data, we can place in the glycoproteins at their positions. This is the neuraminidase, that was the hemagglutinin. We can build in the lipids and the ion channels, and we actually can get these faithful atomic level reconstructions of biological systems. And so why do we want to do this? Well, it does give us, so I would say that when you build a system in this real sense, it completely changes your ability to have sort of the level of insight that you can have into a system completely changes, because now you can see packing effects, you can see neighboring interactions, etc. It gives you an improved sense of the physical arrangement of the entities in your system. It also enables us to study simultaneously multiple components, which is also very powerful. So instead of just having to set up individual simulations and look at everything individually, now we have a sense for the whole. The other thing I want to impress on you is that once we build these structurally accurate systems, we have a platform. We don't have to run molecular dynamics on everything. We might not want to know the wiggling and jiggling of atoms for every system, but we can use this same constructed system to use other types of approaches like Brownian dynamics, which is rigid body diffusion, or stochastic approaches like Monte Carlo-based approaches like M-cell. And I would say all of these are leading us to new avenues of investigation that are not possible on the single protein scale. So for the flu in particular, what we've been able to do, we built it at the atomic level. This is about 100 or 110 nanometers in diameter. It's the largest biological system ever simulated with molecular dynamics. It also has it's sitting in a box of water with ions, but I don't show you that because otherwise you can't see the virus itself. And from a computing standpoint, we were able to sample about 5 nanoseconds per day using a pretty good chunk of the Blue Water supercomputer, and we were able to sample 158 nanoseconds of total simulation, which is not a very long time, right? So we're thinking about biological time scale, and you think 160 nanoseconds, great, Romy, but what are we going to learn? I'm going to tell you what we're going to learn, but before I do that, I want to show you what it looks like. So this is what the trajectory looks like. So you see, we're sort of panning around looking at the system, And I'm rotating and translating the molecule, but also you can kind of see, it'll be easier when we zoom out again because the resolution looks weird. You see the wiggling and jiggling, the very slight wiggling and jiggling of the glycoproteins and the atoms therein. That's the wiggling and jiggling predicted by the MD simulations. So this is not just a video game, this is not just graphics. The power of these approaches is that MD simulations, what I sort of didn't tell you but I glossed over before, is that they're basically something called numerical statistical mechanics. And so we can extrapolate out to experimentally observable, different experimental observables in the lab, like binding free energies and so forth. Okay, so we did all that work. What did we learn? One interesting thing that we learned is that, and here I'm showing, I'm attempting to show you in a very sort of broad way what we call principal component analysis. So I'm looking at we're looking at the hemagglutinin and neuraminidase glycoproteins, and we're comparing what these different heat maps look like between the single glycoprotein simulations and the viral simulations. And just what I want to show you is mainly just the differences between the single glycoprotein and the virus. You can see that actually the glycoproteins in the virus are actually sampling different conformations. And this is in some sense not surprising, but actually it was surprising to some people in the field because I would say it's not surprising because it actually, now what we have is, as you saw in that virus, some of those proteins are pretty tightly packed together. And so what this means is that they're actually changing the sort of the neighboring interactions actually does change the dynamics somewhat. Okay, so then we started looking more globally, and if you just look at the first frame of this simulation, which is shown in sort of a solid color, and I've now overlaid the last frame of the simulation. So I take our time equals 0 and our time at 160 nanoseconds shown in mesh. And you look at that and you think it looks basically the same. And it does, and there weren't very many large structural rearrangements, and to be honest, we wouldn't have really expected that on the order of 160 nanoseconds, so that was okay. What we didn't anticipate at the time, which now I think really is going to catalyze a phase change in how we run and handle biomolecular simulation, is that when you construct these accurate large-scale systems that have many molecular pieces repeating, what we can do is we can leverage the power of Markov state models, which is a statistics-based analysis method, essentially, of our trajectory. What that means is that all those different copies of the glycoproteins, we can basically extract out the dynamics of each of those individual glycoproteins and put them back together into a network of states. And in doing so, the power of this is that, it's that we sampled for 160 nanoseconds, but we sampled that for hundreds of copies of these glycoproteins. And so now, if we're intelligent about how we are analyzing the data, we can actually extrapolate long timescale dynamics from these complex but short timescale simulations. And so what do we learn? So in the case of, so now we have the opportunity to characterize very rigorously the kinetics and the dynamics of any component in our system that we can break down into this framework, which is basically any set of components. So neuraminidase is a long-known drug target against the flu. I'm showing here a picture of the so-called ectodomain of neuraminidase. This is Tamiflu. You probably heard of Tamiflu. Maybe some of you have taken Tamiflu. It's the only orally available drug against the flu. People in influenza tend to care about this thing right here called the 150 loop because it's been shown in some of the in some strains that it's actually adopting an open conformation, and when it does that, there's a pocket down here that could be exploited for drug design. What we were able to do with these simulations is to actually characterize the kinetics of this loop motion in a rigorous way. It also, if we look at the structures coming out of the simulations, we also see, so what I'm showing in the B panel here, in this lower right-hand panel, this is what the crystal structure looks like in green. I'm showing the surface of the crystal structure, and this sort of shiny silver stuff is showing sort of the druggable pocket for neuraminidase. And actually here in panel A is one of the metastable states that we predicted from these long time scale simulations. And you can see a deeply invaginated pocket here, which may indicate that, again, more opportunities for structure-based drug design. How are we doing on time? Okay, I'm just about to wrap up. So, of course, as I mentioned, we don't always want to run and worry about the wiggling and jiggling of every single atom for every single femtosecond of time. And so we don't have to run MD on everything. We can also use these structurally detailed systems to investigate things at the next level, for example, with other types of physics-based approaches like Brownian dynamic simulations, which basically allows us to look at sort of rigid body diffusion of molecules. So if we have our influenza virus and we have sialic acid or different drug molecules, we can actually see how we can test essentially how sialic acid, how fast it will bind and with what preference to different sites on the virus. And so this is really sort of a multiscale approach that allows us to be highly detailed where we need it to be, close by in the receptor binding site, so we can actually get down to the level of atomic level interactions, but acting in the mean field of the electrostatics from the virus. So that the virus will basically steer the sialic acid or the drug molecules to these different sites where they ultimately have these interactions. Okay, so again, why do we care about that? Well, this, for the example with influenza, we can actually we've reconstructed a series of these viral strains mimicking what they see in the field. So we have the 2009 swine flu, we have the 2004 avian flu, and then other lab-created strains. And we can start to investigate things like, for example, one of the things that they see in some of these strains is that neuraminidase, so this is shown, so this is the hemagglutinin, it kind of looks more like a peanut, and neuraminidase looks like a piece of broccoli. So, and over here, here's the broccoli, here's the peanut. So what you can see is that in some of these strains, the neuraminidase has a stalk deletion, so it's much shorter. And so, people aren't sure why, what's the effect of that? Well, we were able to use these diffusional simulations to see how the change in height, which vastly changes the electrostatic driving potential of the ligand to these different sites, how that affects things like kinetics of binding for different host cell receptors. And in the case of influenza, what it revealed to us was an unexpected role of a site on the neuraminidase called the secondary site. So this is a site that has what they call hemagglutination activity. So it behaves, it's a particular site on the neuraminidase that sort of behaves like the hemagglutinin, but we didn't know why. We think actually it's helping to drive. Our studies suggest that this alternate site is actually the first point of contact for flu strains, and it could be helping to drive association and integration with the host cell. And then I think I'll just probably, since we're getting to the end, I'm going to pass this. But we develop also new multiscale methods that allow us to, again, we try to have the best of all worlds. So, and we do this by combining standard Brownian dynamic simulations at distances far from the receptor with very expensive, sort of elaborate MD simulations close to the receptor, and we can combine these two simulation regimes using milestoning theory. And when we do this, we're able to predict the kinetics of binding, and not just binding, but also unbinding. So trying to, again, sort of innovate in the space of developing new methods to quantitate residence time. So drug residence time is something that we are increasingly aware of as being tied to clinical efficacy for drugs. And this approach that we've developed, this new method that we've developed called Seeker, allows us to quantify association rates, dissociation rates, and also free energies of binding, and to do so in a very sort of computationally efficient way. And just closing, as I mentioned, I'm not sure I mentioned, so I know, John, you said that my support started at the NIH when I was a postdoc, I was an F-32 fellow with Andy McCammon, which is true. But I also had a very formative experience in the P41 resource of Klaus Schulten. And there I learned something that I still hold dear to my heart, which is not only all this stuff with the beautiful complexity of the simulation and the intricate piece parts of cells, but also that whatever we do, we try to do it such that others can also use these approaches. And so I have ingrained in me that we disseminate the technologies and we try to train people and train the next generation, inspire the next generation of researchers as well. And so all of the packages, everything that we develop, we do either in collaboration with outstanding established community codes, or we try to develop and disseminate ourselves in robust ways. And we make our software and tutorials available on the website. And we've also been able to develop fairly novel, I would say, technologies for training and sort of recapitulating in-person workshops, but doing that as online courses that students and trainees can watch on their own time. And these include things like virtual machines so that you can actually run exactly what we run with the same data sets successfully on your machine without the headache of trying to do all the installs. Okay, so I hope I leave you with a sense that even over the last 5 years, we've gone in the sense of molecular simulation, we've gone from really being very focused at the single molecule level, which is on the order of about 10 nanometers, to being able to build and construct and faithfully simulate on the order of 100 nanometers. We now have all atom models on the scale that are approaching the scales of a micron and beyond. This is actually preliminary data involved that's showing the actin gel in a dendritic spine, and we can now look to see where the electrostatic potentials, how it's driving calcium release, and all sorts of interesting things. These approaches are really readying us already for the exascale, and we're looking forward to more exciting discoveries with our collaborators and hopefully people like you. So with that, I'll end. There's so many people to thank. I have a fantastic lab and a fantastic summer program. We bring in every summer about 15 high school and undergraduate students. Some of them actually are here in the audience. And that's been great. Of course, I've been really fortunate, I feel, for all the support from the NIH and my mentors. And with that, I will end and take a couple minutes for questions. So thank you.
So we do have time for questions. I just want to remind everyone that there's going to be an FAES-sponsored reception right after this in the library, and everyone is invited. So thank you very much.
Thanks. Questions? I see one.
Yeah, with respect to your work on P53,
yeah.
There are animal models such as the elephant and the blind mole rat who have P53 alterations which are beneficial in terms of combating cancer, and that neither of these mammals, large or the small one, get tumors very often. Have you considered looking at their P53? I know the elephant probably has more genes for P53. The blind mole rat has a different type of mechanism. I forgot exactly what it is, but those seem like they would be of great interest in your quest to define
what's happening,
beneficial things for P53.
That is a great suggestion. That's a great suggestion, and actually we haven't looked very much into that, but I can assure you that when I go home, we're going to look right into that. Yeah, I'm actually, we haven't looked much at those models, and I know, but that's an excellent suggestion. So we'll do that. Thanks.
Thank you.
Yeah.
Hi, I don't know much about molecular dynamics, but I just wanted to know are your models entirely classical, or is there any quantum mechanical, anything involved?
Thank you for asking. So the question is, are they quantum mechanical? No, they're not. There's no bond breaking, there's no bond making. So what we put into the system, the bonds that we initially put into the system are the ones that stay there through the system. If we wanted to look at reactivity, reactions, we would have to do something like QM, MM, which is possible, but not in what I described today. Yeah. Thanks. Yeah.
So I have a question about your Markov modeling.
Yeah.
So when you try to see some long time scale dynamics from very short time scale dynamics, basically you don't have much information what happens in very long time. So I think if you want to model using your Markov model, then you just repeat the short time information repeatedly. So is there any criteria, if, for example, if you want to see some microsecond dynamics, then how long should you model from your MD simulation?
That's a great question. Okay, so one thing to clarify is that you don't actually need to sample a full transition in any one simulation. That's the power of the Markov state model. That you can locally sample and resolve transitions at a local level And then you sort of bridge them together through this elaboration of states. So what we and others have shown is that, you know, you can get, basically it goes out, and it depends also, so it's a little bit of a tricky question because it depends how you run it. So there have been studies by, for example, folks like Frank Noe, who I think have done microsecond type simulations individually, but actually can extrapolate out all the way to seconds and even possibly minutes, and that's because they're using enhanced sampling approaches on particular parts of the space that are otherwise very difficult to sample. So we can talk more about it afterwards. I don't want to get too overly technical, but like for the example of neuraminidase, I can tell you that you can sample on the order of nanoseconds, and you can extrapolate out to millisecond timescales.
Thank you.
Yeah, if you do it properly.
So I have two questions you can answer, which were one with respect to time. So the first question is that with respect to druggability specifically, is there anything that you learn from embedding let's say a protein in one of your larger environments that you don't learn by doing the isolated MD simulation in its immediate environment? The second question is that as you do this sort of large-scale model building using experimental information from multiple experimental systems, how do you think that you are going to essentially assign weightage to the different constraints between the different experimental systems which have different levels of accuracy? And as people are downloading these and making their make own models, how do you envisage training them about that?
That is a great question. Okay, so I'm going to try to answer both questions. I'm really bad with answering questions in succession. So the first question was about what can we learn for drug targets in the big systems that we couldn't learn in a single molecule study. There I would say one of the really interesting things that we've been able to discern and learn about is when we're looking at diffusion of drug molecules to their site of action, we can actually see where else they might be going. And if you think, so I think that, ultimately we might get a better sense, at least in the sort of local environment, where else, and, and it depends on the scale of the model that we build, but ultimately it would be great to have detailed enough models that it could really sort of more accurately inform on off-target effects in sort of new ways. That's the answer to the first question. For the second question about data, essentially data reliability and reproducibility and how we...
And maybe building like an equivalent of an R-free that we use in crystallography for systems such as these.
Yeah. Yes, that's a very good question. It's a very tough question. So I think that a lot of the educating of the population obviously has to just happen in the labs, in the ether, that we don't have perfect reproducibility, even when the constructs are the same. You pass it to a different lab, sometimes, things can change. Any number of things can change. And so, yes, it's something that, at this point, it's nothing that we do in an automated way. And I think, as you know, this is like just a big challenge for science in general, and trying to, either using, for example, I know I was just at NIST yesterday, and they were talking about how they're developing different capabilities to put sensors on various parts of the lab so they can monitor, all sorts of different conditions that might be changing that people don't even think about, and then reporting and recording and so forth. But that's a whole different conversation. But it's something definitely that we have to be careful with. Other questions?
So let's thank Romy again for a wonderful talk. Thank you.